mucho ruido estático. Hola, 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 hola.
Hello.
Hello? Yeah, can you hear me? Is it on? Yeah? Okay. Excellent.
Hola. 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 Yes.
Speak loud. I won't whisper.
So, good morning, everybody. So, let's start this seminar. So, it is a real pleasure to me and to the institution to receive Dr. Andrew Filby, but he likes to be called Andy. Only his mother called Andrew when she was angry at him, so he likes to be called Andy. So Andy Filby likes cats, long walks through the forest, and also likes chocolate.
Yeah.
And apart from these nice things, he's in charge of the flow cytometry core facility at Newcastle University. He did his PhD and postdoc at the National Institute of Medical Research, and working on mouse knockout models and TCR signaling on retroviral infection. He's very active. He likes it very much to train and teach people and to distribute all his knowledge. And always he is working and trying to understand on cutting and develop cutting-edge cytometry and all areas related with this. He has a lot of publication. And recently he contributed with the Science publication in 2007 that is the first recognized output from the Human Cell Atlas initiative. So it's welcome, Andy, and we are happy to have you here.
Okay, so thank you very much for the wonderful introduction, and again I'd like to say thank you to Mariella for inviting me. I was here in 2014, wasn't I, the first time, so almost 4 years ago to the day, actually, or the week. So the problem is, though, that I almost didn't make it. I don't know if you've been following the news, but the United Kingdom was very heavily attacked by this thing called the Beast from the East. Yeah. Now, to tell you where I'm from, so I'm from Newcastle, so I don't know if you know your UK geography, but you know, here we have London, of course, and then Scotland, but Newcastle is here, right? So if you see, this is where the heaviest snow was last Wednesday. So poor Mariella and also Fernando from La Plata were getting kind of almost daily updates or hourly updates from me about whether I thought I could make it. But then there are some people, this is my daughter, so obviously the snow made her happy, but it made me very worried because this was the view from Newcastle Airport and you can see the snow in the background. However, Mariella is connected, she knows people, and she said she knows a guy from Buenos Aires who's got a bit of power and a bit of sway. So obviously she had a word and she told me that the power of South American prayer is what's going to get me here. And there's no doubt that if I wasn't a believer in South American prayer, I am now. Okay. They can make planes on time. All right. So, right, okay, I'm going to return to being serious just for a bit. So this is the overview I want to speak to you today about. So I want to kind of frame this idea of cell heterogeneity, the idea of identifying what your cells are, who they are, and what they do. And of course, you know, as Mariella said, I am a cytometrist, so therefore I believe very strongly that cytometry is the way in which we do this. So how do we establish cellular identity? Well, when it comes down to what we do, we make measurements on cells. We derive parameters, and we want those parameters to have power in resolution. And I don't mean resolution in terms of image quality, although that is linked. I mean resolution in terms of pulling cell type A away from cell type B and distinguishing identity. Okay, so I'm going to speak a bit about that. And then the data I want to talk through, two kind of areas of work that we've done over the last few years. This is the stuff that's very well published now, and we always joke about this. We feel that we've taken this data and kind of rinsed out everything we can get from it. But then I also want to talk a little bit about something new at the end. But what we've been doing is we've been using what we call label-free cytometry to look at cell cycle in mammalian cells and also in yeast cells by this technology called imaging flow cytometry. I'll introduce that. So I'll talk you through some of the challenges we've had with the data analysis, where we've tried to begin by kind of being rather manual in the way that we do it, but then I'm also going to kind of bumble my way through this field of deep learning that we've been working on recently to try and extract, again, parameters and resolution and establish cellular identity. And then at the end, for the last kind of 10 minutes, hopefully, if Mariella's not going like this at me, I want to talk about this new project we have where we call it label-free CDs. So if you're an immunologist, you know that CD markers are what we stain for to establish cell identity, but we wonder whether we can refine or replace the need for CD markers with something which is actually a label-free feature. And then we'll finish there. Okay, so again, I think hopefully everyone in this room would identify with one of these questions or problems, right? So heterogeneity for me is one of the biggest issues. Not only are we heterogeneous as a group of individuals, but the cells that make up our bodies are highly heterogeneous. Now, if you work on a multicellular organism, I think I'm not going to have to convince you too much about cellular heterogeneity. You know it exists. Specialized cell types performing their functions, not doing their functions, are what really give us our health and disease, okay? But I also want to point out that even cell types such as cell lines or bacteria, yeast, communities that you think are homogeneous are not. They're not, because a very good example is transition states. Cells that are going through the cell cycle are transitioning. They are heterogeneous. Here we have an example. We have cells in G1, S, and G2 just on DNA content. These cells are not the same. In that particular moment, they're not the same. The other question we often have as well is that two cells may look the same, but they may be doing something different from one another. A good example is a cytotoxic T cell may look phenotypically similar, but it's not doing its job until it's actually in contact with another cell and killing it. Okay? And then the other one as well, which I've become very interested in recently, is this idea of location. The spatial context of a cell type in blood or in tissue could also influence its behavior. So all of these things are a challenge when we're trying to understand cells in any system. Okay? So establishing cellular identity is definitely a fundamental question to establishing health, disease, and just understanding your basic biology as well. So as Mariella said in the introduction, hopefully there is knowledge and understanding of this very ambitious project called the Human Cell Atlas. The mission is very clear for me and very, very, very grand and extremely worthy. It's to create comprehensive reference maps of all human cells effectively in as many individuals as possible, and I guess in as many settings as possible as well. And I'm very fortunate enough through my work, my place of work, to have been involved in this great study, which I'm not going to talk about today, but I just wanted to highlight it. This was driven by Nir Hakkinen, Alexander Chloe Veleni from the Broad Institute, but also through my workplace, Professor Mazlifa Hanifa. And the question here was to really use novel and powerful cytometry techniques, single-cell RNA sequencing, to look to see whether you can define a cell population by effectively asking more questions and more parameters. All right, so how do we begin to prove cell identity? This is what I do. I look at cells and I want to know what they are and what they're doing. So the answer, again, for me is this. It's cytometry. And you know that cytometry, it literally means cell measurement. You don't have to define it any more than that. But this is my daughter again. She's going to feature. She likes to feature in my talks. But this is my daughter doing an experiment. She's looking at testing the pH of different household chemicals, like washing liquid, and using this extract from the red cabbage. But you can tell from her dress, she wants to be a scientist, and she asks me a lot about what I do. So when I tell her what I do, I say, I ask questions. I ask questions about cells. I want to know what they're up to. So she decided she would come up with this term that a cytometrist is a cellular detective. We are like detectives. We have a crime to solve, and we have evidence, and we have suspects, and we want to drill down into who is doing what and where, right? And then this is, again, very relevant. I put this in ages ago. I use it all the time. But, this game here, this game of Guess Who, this was a favorite game of mine when I was a kid. I worried when I made this slide that maybe something would be lost in translation. So maybe that's fair. Yeah, that's better. Okay. But this is it in a way. As cytometrists, we're playing this game of Guess Who. We're saying, if we're immunologists again, does it express this? Yes. Does it express this? No. And this. And at the end of it, you make a conclusion about what it could be. But hopefully you can see that the more questions we can ask, the more understanding we have. And the problem is as well, some of the questions might be important and some are not. But you only know who's important and who isn't important once you ask those questions. And this is always the challenge for me. So what do we ask? Let's talk about these questions that we would ask ourselves. If I was told I've only got 4 questions generally to ask of my cells, these would probably be the ones I would ask. So yeah, you can tell this cell's in trouble here. Right? So I would want to know, what do they contain, express, or produce? What do they do? What do they look like? Okay, this is what I'm going to talk about. I'm going to talk about imaging cytometry. Where are they and who do they associate and not associate with? So I just want to highlight those 3 and 4 questions because, again, hopefully you can tell that I can't really do this without seeing an image of my cell. And the other thing as well, I mean, I believe that cytometry is such a massive subject, a massive field. And, you know, I don't just believe that it's restricted to looking at receptor expression. This is something we often think of when we think of cytometry. We add our antibodies onto the surface, we run them through a flow cytometer, and we say this is positive, this is negative. But we know as well it's not too difficult then to look at things like ligand binding on the surface of cells, looking at signaling cascades, looking at phosphorylation, but we can also look at DNA. We can look at DNA modification and DNA content. But obviously, most recently now, we are apparently in the dawn of the single-cell analysis. And again, as a cytometrist, I see the irony in this because flow cytometry is the original single-cell technology. You have your cells in suspension, and you measure them one at a time. Now, of course, it's very impressive that we can now look at RNA. We can look at the transcriptome of a single cell. But I still view this as cytometry. And I know this is being recorded, so I hope I don't end up being excommunicated for this. But I believe that single-cell RNA sequencing is just another form of cytometry. Okay? But the important thing is, and this again is cytometry, is yes, we measure one cell at a time, we measure as many things as we can, but what we're trying to do is we're trying to take that information and put it back into the population context. It's not about just measuring a couple of cells, it's about measuring as many as you possibly can, because we want to sample everything, right? Okay, so how do we ask these questions, right? So, bit of a busy slide here, but imagine here's our cell here. You know, how do we ask the questions? Well, traditionally, we use probes and labels. Now, antibodies, I don't have to introduce those. We can look at antibodies, use that specificity, put different analytes on the back of these antibodies, and ask questions of our cells. We have chemicals we can use genetic tagging with fluorescent proteins, but we can also use oligos as our probe as well. We can put analytes on oligos. And then one of the things I really enjoy about what I do as well is that we often have a lot of functional reporters, things that tell us about the way the cell's behaving, fluorescence dropping with proliferation or fluorescence changing with calcium flux. But this is what we want to do. We want to paint our cell or introduce something about our cell that we can identify. Okay, so I want to go into the technology now. So, you know, cytometrist, cellular detector. Cytometry is cell measurement, so we do this using cytometers, the instruments. These are our tools of our trade. So I'm very fortunate in Newcastle that I have a very well-funded institute that has a lot of good technologies available to us. So I tend to group them into 2 groups, and then I like to also comment on this idea of how many measurements they can make per cell, and also the idea of how many cells they can measure. So I find I like to group things. So we obviously have our conventional fluorescence-based flow cytometers. We have the, in fact, Symphony system, which allows us to make 30 measurements per cell, so we can ask quite a lot of questions. We work as well with mass cytometry. Now, mass cytometry is where we leave fluorescence completely alone, and we use metal-tagged isotopes as our analytes. This allows us to make more measurements per cell because we do not have the crowding of the fluorescence space. But we do this in suspension. Okay? And then more recently, not particularly under my care, but we have the availability of this incredible 10x chromium system from 10x Genomics, which again is a bit like a cell sorter, and this is particularly used for looking at single-cell RNA, preparing libraries of single-cell barcoded RNA. But again, this doesn't give us an image of the cell. Okay? We don't know anything about the shape or the size or the spatial location. So we also work a lot in the image-based cytometry field. So we have the system that I'm going to really focus on today, this imaging flow cytometer. But more recently as well, we have a module which sits in front of the Helios mass cytometer, which allows us to image tissue with highly multi-parameter reagents, again, metal-tagged antibodies. So this idea here is that we move from, you know, kind of a lower parameter space to a high parameter space, but there is always this kind of inverse relationship, unfortunately, with speed and throughput. But what's important is all of these systems can give us this multi-parameter data at all these different levels of cell biology on an individual cell basis. So we can use them for looking at mRNA and protein. So this is the focus. Let's start on this. So the imaging flow cytometer was made originally by a company called Amnis. Amnis were from Seattle. I've been very fortunate to be involved with the technology from quite a long time, about 10 years now. So what I want to kind of do is I want to just give you a little bit of an overview how the system works, because if I'm going to talk about the data from it, I want you to understand how the data has been generated. So here is the schematic layout of the image stream system. So let's highlight some stuff here. First thing to say is it's a suspension technology. So if you work with liquid suspension cells, that's kind of easier. If you work in tissue, you have to disaggregate into a cell suspension. But we have the flow cell. There's the flow cell. So the flow cell, just like in conventional fluorescence flow cytometry, is there to push the cells or particles through the laser beams and basically excite the molecules that are on our cells. So in order to do that, again, we have lasers. So we have excitation. This tells you, again, this is fluorescence-based detection. But the thing that I'm going to really focus on today is we have bright field as well. We have the ability to place LED light, transmitted light through the flow cell to obtain a bright field image of our cell. So once we excite those cells, produce photons, produce light, it then goes to a filter stack. Now the job of the filter stack, again, is a set of mirrors, long pass mirrors. Each one chops light up, emission light, into a particular wavelength. And then finally, those mirrors are actually angled to place light of a given wavelength onto a CCD camera. So we have a spatially registered detector, a CCD, made of pixels. Now the system I'm showing you here is a two-camera system, and what that means is, if you can add that up, it's 6 channels per camera, so it's 12 images per cell in flow. So I want to talk a little bit how this works, because I do think it's very, very cool. It's very clever. We are imaging a moving object. So this object is not static, it's moving. So the way this is done on this system, it uses a technology called time delay integration. So in this cartoon, I'm going to try and explain this. Here we have our excitation laser. Here we have our cell that's been labeled, moving down into the excitation laser, into the flow cell. We're then going to have light in this cartoon generated that's going to be decomposed into its spectral elements using this filter stack, and then each part of this image is going to be transmitted onto an example of 3 channels on the CCD array. So let's see how good my animation is, right? Okay, so cell comes into the beam, it's excited, the light is broken up, passes down the camera. What happens is that the information from the camera on these pixel rows is passed down at the same rate the object is moving. What this means is it's digital panning. So instead of the camera moving with the object, the information moves with the object like this. Now there's several benefits to this. One real benefit is that actually as the cell moves down the camera, integration of signal continues. So what you end up with once you pass down the camera is your fully integrated spatially registered image. And this is how we can get very good sensitivity on this system without boosting signals using PMTs and things like that. Right? So this is how it works. And this is an example of output. So this is just some nice pretty images that I'm trying to throw up here. Oh, thank you, Maria. There we go. Don't fall asleep. Right? These are some nice images showing some cells which have been labeled up with antibodies in exactly the same way as you would label for fluorescence, non-imaging flow cytometry. But what we're seeing here is we're seeing the output. So this is a cell, and we're looking at its bright field in one channel, a repetitive bright field on the second camera, and then we're looking at the fluorescence information all the way across for that one cell. So you can imagine here that, if we have lots of cells running through the system, this is just going to go on and on and on. But this is what we get. So 12 spatially multispectral images per cell. And again, I want to stress the point that this is imaged a moving object. And you may look and say, okay, the image quality is not great, but this is where the beauty of what we've been doing with the image analysis comes into it. It doesn't matter, okay? All right, so that was serious. So let's become less serious, but with a serious message. So this quest to try and find the technology which gives you the most measurements on the most cells, I jokingly decided to call this the parameter space race, right? Because everyone's kind of questing for this ideal technology, and I think it's a good thing. We'll get there. We'll get there. But I think it's great to have this idea that this parameter space race could actually have some interest within the highest powers of the political forces in the world. So this is a really real picture of a few years ago when Barack Obama, when he was president, visited the NIH virus unit, viral research unit. Now, he looked at the machines, and if you go on YouTube and you Google Barack Obama and flow cytometry, you see that he gave an announcement to the NIH afterwards, and he pretty much described what it was. So he gets it, right? He gets everything. This guy, he's so cool, right? But the problem is things have changed, right? So I was worried about this, actually. I'm a bit worried. I hope I don't end up being the target of abuse from this man on Twitter. Let's see. But, of course, President Trump, he's going to want to do it better. So he says here, this Obama guy thinks he knows all about flow cytometers. So I bought the biggest and the best with 7 lasers and a gazillion detectors, right? Because that's exactly what he'd say. And of course, Vladimir here says, and again, I worry about this, Vladimir, if you're watching. Good job, right? My Cytof Helios can measure 40 parameters, and Russia owns all the isotope mines. So now, this guy, hopefully you know who this is. This is the President of France, Macron. Well, you know, I think we can all agree, what a smooth guy, right? He's smooth. But this is where Macron, he says, well, actually, gentlemen, I think you find where cytometry is concerned, image is everything. So I'd like to thank the world powers for commenting on cytometry. Right? OK, so again, I want to put this into a cartoon. So in this cartoon, what we've got here is we've got the number of single cells that are practical to measure. Not theoretical, but practical. And then the number of parameters that we can measure per single cell. So this is where I like to put flow cytometry. It's extremely high throughput. You can get hundreds of thousands of cells through very quickly, but the number of measurements we can make is still restricted by the fluorescence. I think that will change, but at the moment, that's about where we are. Mass cytometry in suspension gives us a bit of an uplift in the number of measurements we can make because we're looking now at metal, not fluorescence, but this is compromised by the speed of the system. It's slower. Again, this is probably out of date because this changes on a weekly basis, but for me, single-cell RNA sequencing, of course, gives you this ability to look at loads of things per cell, 2,000, 4,000, 6,000 transcripts per cell, but the throughput is still a little down. Okay? So I place imaging flow cytometry here. I place it as having very good throughput and even more potential parameters than single-cell RNA sequencing. And again, this makes me a bit unpopular, right? I have to justify this. I'll just quickly make a mention of tissue cytometry. Again, I like tissue cytometry because, again, we're not looking at suspension cells, we're looking at cells within the tissue. Therefore, not only do we get all the other information, we also get the localization as well. So I believe that this moves our parameter space up here. But this is a question I'm always, where will we be, how will we get there? But that's for another talk. But here's another political figure. This is the genius that is Donald Rumsfeld. And this quote was taken from when he commented, I believe, on the search for weapons of mass destruction in Iraq. I don't know, right? So he said this. He said, there are known knowns. These are the things that we know. Right? There are the known unknowns. That is to say, these are the things that we know we don't know, right? But there are also unknown unknowns. These are the things we don't know we don't know. Now, people saw this and thought the guy was, you know, maybe not all okay sometimes. But I think this really comments beautifully on what we do. We as cytometrists, as cell biologists, we want to know, we want to find the unknown unknowns and the known unknowns. But we also sometimes want to find the known knowns to know that we haven't found something that's completely crazy. But I think Rumsfeld language is great for describing what we do. But this is what I see. I see these kinds of technologies very much in the discovery area where you can find new things about your cells. But the problem is you have to validate it. Two reasons you validate. One, if you're looking at single-cell RNA sequencing, it doesn't mean if the RNA is there, it doesn't mean the protein is there. So you need to find a way to validate that. The other issue as well, if you're looking for a rare cell population, you need to have the throughput to be able to find, as we say, the needle in the haystack. So I think we're in an interesting position. OK, so let's go back to imaging flow cytometry. How can I tell you that it's better than RNA sequencing? How can I do that? I've only got 12 channels of information, right? So how can I say this is a world power in this parameter space race? I think, you know, maybe you should be thinking he's crazy. But the reason is this. It's because we have digital imagery. That's why we're generating CCD-derived digital image. So for every image, every channel, every cell, we have pixels, X, Y coordinate. But more than that, we have a dynamic range to those pixels, a detector. So in this case, I work with a CCD camera. It's 12-bit. So every value is digitized between 0 and 1496. So now I don't just have my X and my Y projection, but I also have this idea of texture in the image. And again, all of you, I'm sure, many of you do image analysis, so I don't want to labor this point, right? There's more information in this than just an X and a Y, okay? So what this means now is, for me, there's nothing more information rich than one single digital image, except maybe lots of digital images. So you can take this information and we can do intensity-based features. We can look at some intensity, but we can also look at the shape of the projected image. We can look at circularity, aspect ratio, these kinds of things as well. We can look at texture, and we can look at things like internalization. So this is just a tiny, tiny, tiny snapshot of what we can derive from one image. So now think to yourself, I now have 12 images, 500,000 cells, and you start to see the challenge and the power of what we can find. Okay. But what about this? What about label-free? This is the point of this talk. We'll get there in the end. Bear with me, right? The point of this talk is to think about how we can ask questions of cells without having to add label. So we know that conventional fluorescence flow cytometry gives us two pretty useful label-free parameters, maybe three, actually. We have the forward scatter light, we have the side scatter light. Also, we may hate it, but sometimes autofluorescence can be very useful too, right? Beauty of this system is we get all that. We get side scatter, autofluorescence, but we get the transmitted light feature. And this, just to emphasize the flashing there, this is kind of unique. It's not unique to microscopy, but it's unique to flow cytometry. Right? So this is it. Let's get into the data now. So looking for the hidden features in transmitted light. This is what we want to do. I believe transmitted light imagery is very powerful. I believe that it tells us things. I think there are things in there. So I don't know if you can see this, but this is a real cell. Didn't do anything to it. It's got a kind of a weird, evil smile, right? And then this is definite, and you have to bear with me. This looks like the helmet from Darth Vader, right? Yeah? I'm not crazy. This is definitely the helmet of Darth Vader, all right? Okay, so I want to start with a worked example. As I said in the introduction, I want to talk about the cell cycle. This is what we used this technology for. So I don't have to labor this for you because you'll understand the mammalian cell cycle, but effectively it's made of phases. Each phase is responsible for preparing either the cell for division, for replicating DNA, or then preparing cells to divide to produce two daughter cells and then maybe go back into it again. So I don't need an image to get a load of information. Just conventional fluorescence-based flow cytometry has always been very powerful for looking at cell cycle because we can just look at propydium iodide intensity, and maybe if we want to look at mitotic cells, we can add a mitotic marker as well to break down G2 and M. But for reasons I won't bore you with, we were obviously very interested in pulling more out of this. So as we know, M phase is a really heterogeneous population. The cells are moving through pro, meta, ana, telophase, and of course then back into the interphase. So we realized that imaging flow cytometry would be a great way to get into this, right? A really nice way to try and not just look at the gross cell cycle phases, but actually get into the mitotic phases as well using nuclear morphology. But then the other thing we started to think was, well, you know, what could we do with these images? Maybe there's something in there that will remove the need to do any of this staining, okay? So we began, obviously, as like children in this. We wanted to do it manually. And I'm going to talk a little bit about some of the challenges there. So this is me. This is a statue of me worrying about how to do this, all right? And it's published as well, so again, you can follow this afterwards. You read it at your leisure this afternoon with a matter or something, and yeah, but this was published in 2011, okay? But what we did was this. We have the system. We run our cells through it, labeled in this case with propydium iodide and with a marker to identify mitotic cells. And we just treat it initially like a conventional zero-resolution flow plot. We have an integrated image intensity here in both these channels, and we identify the cells that the fluorescence tells us is positive, is mitotic. But then what we do is we then look at these images and we start to interrogate them. So we start to say, okay, what is it about a prophase cell that could make it different from a metaphase or an anaphase or a telophase? So what we said was actually, well, I don't know if you can see this too well, but this has one nuclear foci. So maybe we'll do a spot count in the nucleus, and it's kind of round. So maybe we'll look at a feature which tells us about roundness. Metaphase, still got one nucleus, but the aspect ratio now is starting to elongate, so, or the size is elongated, so the aspect ratio starts to drop down. And then telophase and anaphase should give us a spot count of 2 nuclear foci if we get things right, and then an aspect ratio of this. So that's great, but what about, again, this idea of being label-free? So the idea for me is that label-free could be quicker because we don't add any staining. It could be cheaper because we don't have to buy some company's expensive reagent. And then, as I'll talk about hopefully at the end, it's maybe less likely to alter the biology of what we're doing. We like to think that when we add a probe to a cell, we haven't changed anything. But if you work in immunology, you'll know that adding an antibody against the surface receptor mimics a signal, and it can change things, all right? So just a little emphasis there. So what's in the bright field? So as a human, I think I'm a human still, after this week of 6 talks now, the human eye can easily discern a qualitative difference in something, but we need to enumerate. That's what a cytometrist is. Cytometry is not about looking at 2 images and going, well, that one's a bit more than that one, or this one's a bit different. We want to enumerate and put a number to this. So I fully accept that a computer can do this, but me, not so much, okay? Right, so let's think about this idea. So no matter what cytometry platform we're using, all the systems I've shown you, for me, any technique, no matter how many measurements you're making, the key to them proving your identity has got to be this. It has to be the resolving power of your parameters that you have chosen or selected to literally describe in numerical terms the difference between, in this case, cell type A and cell type B. I don't think you can get away from this. I really don't. Okay? And again, the parameters might not be one parameter. It could be, n parameters, as many as you need to resolve A from B with the biggest difference. Okay? But what are the enemies of the resolution, right? So in 0 resolution cytometry, where we do not have an image, it's a bit easier to quantify in some ways. So the first thing that would maybe go wrong is you just don't pick the right reagents, you don't pick the right probes, you don't use good reagents to ask your questions. The other one, of course, that you never get away with is the machine's not very good. It's misbehaving. Your measurement is error-prone. You have high background. You're now going to have your signals merging into one another. This one is a bit more interesting. Once you have all your measurements on this system, then maybe you just go about selecting the wrong parameters to pull these populations apart from one another, right? So as I said, 0 resolution cytometry features, they do tend to be a little more, for me, a bit more binary in nature. It's, you are positive or negative. If you're positive, then this is how positive you are. And we always have this challenge because of the limited parameter space, the limited number of questions we ask. Discovery is less likely because of the need for an a priori knowledge and a bias about what the biology is you're looking at. So if we think about image cytometry, we can't get away from this, and certainly don't get away from this, but the problem actually now is we have a greater challenge ahead of us. It's not about being positive or negative for fluorescence, but you also have to think about how you do the image analysis. And these 2 things, like segmentation and then feature selection, are absolutely affecting one another. Okay? So let's put that there. So yeah, get rid of those 2. Let's talk about that. So I'm going to go over this quickly. Image segmentation, what you're doing is you're basically trying to look at the pixel values in a digital image and identify your single cells in a tissue or your structures within those cells, right? Let's not go on about it anymore. But I want to give you an example, a horror story. When we were doing this manual analysis, we realized very quickly that these fancy, clever features that I came up with, the spot count and aspect ratio, of course, were utterly dependent on the segmentation. So here in mask 1, I'm showing you the default segmentation that comes from the software. And hopefully you can kind of see the purple color. We're not seeing, it's not recognizing 2 foci here or 2 foci here. It's also not recognizing this narrowed shape, this elongation of the metaphase plate. So I put them in my feature and I just get 1 population, nothing. I don't pull apart any of those phases at all. If I go back now and I modify my masking strategy to be better, to be smarter, snapping to the morphology a little bit more, then I get my resolution. So this was a problem. This was a problem for us because we wanted to be reproducible. We wanted to be able to push this method to other people, use templates, but we found that we were constantly playing with the masks and tweaking and tweaking. And it obviously required a lot of expert input from me as the person to annotate the data, and this is not what we wanted, okay? So the manual approach was hard because we particularly found it very difficult to find a feature that described or resolved. I mean, it was very much, down to me, my imagination, and that doesn't work for me, right? So it was subjective and, again, analyst dependent and not always repeatable and reproducible. The other thing we found as well was that, you notice we weren't really using the bright field image. We weren't really using it. We weren't getting anything from it. We were relying on the usual suspects, the fluorescence, the nuclear morphology. But, I want to say that no matter which fancy technology, and we'll talk about analysis technique we use later on, manually annotated data by an expert is still very powerful for training classifiers and validating networks, and we'll talk about that in a minute. Okay, so let's get rid of all this now. Let's think, can we eliminate all things, right? So this is where we're going to talk about a little bit about machine learning. And again, massive disclaimer here, I'm not an expert in machine learning. I'm not an expert in deep learning, but fortunately, I get to work with people who are very good at it, and they carry me through just about, right? So for me, the idea of classical machine learning, it will help find best resolving features, But the way we tend to work with it, it still requires the segmentation and masking, right? So we have a problem there straight away. We have a benefit, but we have an issue. Deep learning, as far as we felt it worked for us, it can find resolving features without the need to pre-segment your data. You can just put the data in, just feed it in, okay? So let's talk about that. So I knew nothing about deep learning till about a year or 2 years ago, maybe a year and a half actually. But when I realized, you realize it's everywhere. I mean, it's everywhere. It's in everything. It's in all parts of our lives. And there's some very interesting stories about how it's been used. The first computer to beat a professional player at this AlphaGo. It's also being used a lot in healthcare, in diagnosis. And of course, it's also being used by these guys. Again, I hope maybe the YouTube will just get cut now I've said this. But it's there and it's learning what we do. It's learning what we do. Spotify, things like this, picking the music that we like. My wife's Spotify account is mostly dominated by Disney songs now because, my daughter uses it. But yeah. Okay, so deep learning, benefits and issues. So again, as I said, there's a potential to have no need for manually segmenting your data or feature extraction. There's a possibility as well for a more objective approach as well. There's not going to be biased by my a priori attitudes. We found as well that imaging flow cytometry is quite well suited to this idea of deep learning because we get lots and lots and lots and lots and lots of data points. I believe as well that, because of this high throughput, high content thing, you maybe have less chance of a fear of overfitting something. So having this problem where, you basically come up with a way of analyzing your one file that you tested the model on, but then when you apply it to another thing, it's just nonsense because it's only fitted to the one sample that you put in. My issue, though, and I have a horror story about this that maybe we can talk about another time, but not sure what's been learnt on. That's my little uncomfortable feeling about this, is that maybe it's an abstract feature, maybe it's not truly biological, but maybe some kind of image artifact. And I was very careful to make this distinction because even if it's something biological that I didn't think of, doesn't mean to say it's not powerful at resolving the biology. My worry is that there could be an artifact in the image, but okay. So, completely lifted from Wikipedia, deep learning is a branch of machine learning that's based on algorithms that attempt to model high-level abstraction. And of course, the idea is that it tends to fit with this concept of the human brain, right? So having networks, okay? It's composed of multiple linear and non-linear transformations, right? So, I guess in our head, when I'm thinking about a bright field image that we're putting into this kind of thing, what it's doing is something like a convolutional neural network. It's looking at these different image filters, transforming raw input image into basically a map, a feature map. So it's looking here and just looking and making these little feature maps on this original raw input image. And then this gets pulled, which I also found quite interesting because the idea is that you're actually downsampling the feature maps to reduce the number of pixels, so you're actually becoming even less, I guess, nice imagery. But then what you get at the end, hopefully, is this output layer, which is connected with a network and assigns a class to each input image with a certain probability. So I guess in this example, class 1 and 2 could be leukemic and normal cells, or cells in metaphase or anaphase. So again, I had to think about this a bit more. So I was speaking to my collaborators, and they were trying to give me examples of how you can think how deep learning works. So the one that I liked was this idea of face recognition. So you put in some, you make a network, and you put in pictures of, I think it's a human face, I don't know, and cats, and you hope that the network learns to distinguish cats and humans. And I like cats, and I like most humans, so this is a good way. But then I thought, well, that's okay, but cats and humans normally look pretty different to one another. So what's power in this? So I found this thing on the internet. Basically, there's a whole movement of people that like to wear cats on their head. It's great. So can it still distinguish something like this? And I'm assured it can. So that's the power for me. The power is that you can teach a network, grow a network to find very, very subtle differences. Okay, so that's that part. Let's actually look at some data. So this work was published back in 2017. It's still quite recent. And it's really driven by a really smart guy called Alexander Wolf, who works for Fabian Thies in the Helmholtz Center in Munich. So we fed him our data, and he basically said that I can use a convolutional neural network and deep learning to basically reconstruct the cell cycle and also some disease data. So this is the idea. So we called it deep flow. So the idea again is that you generate your imagery, you then create a network, you have your input images, you have all your convolutional neural network, your masking, your nodes, and then you have your global averaging at the end. And then with the softmax classifier, the idea is you start to try and classify into different phases, and then obviously we can use things like TSNE to visualize the output. So if you inspect the last layer of the network, we can also test it then with our annotated data. So again, it's still important to have, in my view, very good, very carefully annotated ground truth data to then either test the network or retrain it. So this movie, I love a movie. So this is a movie actually of running the network on some known class cells. So I think it's quite nice. So here, you know, in this sort of activation space, you've got all these bright field images that we've got in our data file. You can see that, you know, some of them are not massively good quality. You know, there's a little bit, I wouldn't sort of say it's amazing, but then you run the network, and what you find then is that we visualize again in this TSNE space, is we start to see the cells finding their place in life. And there's some kind of cool things here. You know, you kind of get this like sausage arrangement, and then you also get this little kind of pocket of events which are sitting out here, and I'll talk that through in a minute. So of course, yeah, you can rotate it around and make it look really pretty. But this is what we got. What we got was, again, visualizing this in three-dimensional t-SNE space is not only class identification, but also this concept of some kind of progression. So obviously, we call this pseudotime. So it seemed that based on about 336 feature dimensions visualized using this TSNE algorithm, we were able to basically obtain what looks like a progression of our cells, very much like you get with propidium iodide when you look at the intensity change. Okay. So again, I worry because it's nice to, you know, I don't trust something that just gives me an answer at the end. So one of the things that was done as well was that they went back into the network to look at the different maps. So just to kind of highlight, sorry, I apologize about that. The first map that seemed to be quite powerful was something that looked at cell boundary. They found the cell boundary. Second map there, I think I've got a bit of a problem there. The second map was, I think, local scatter intensities or intracellular size. Oh, sorry, this one was local scatter intensity. And then there seemed to be another one which possibly was a cross-channel feature which seemed to suggest that the network had actually learned how to segment as well, which I think is pretty cool. But what we wanted to do as well was then compare and contrast what we got to classical machine learning data. So what was interesting was that not only did we get this kind of ordering in pseudotime of the different phases, but we had this massive, well, this little distinct population pulled out here. And what these were on further inspection was these were dead cells. These were dead cells that missed my apparent expert ability to find dead cells. So this is nice. This gives us this idea of an unknown known. So I probably knew that there was dead cells in there, but I didn't have the analytical skill to pull them out. If we visualize classical machine learning data using this technique called gradient boosting on the same data set, hopefully you can kind of see here that there is some class labeling, but we're not ordering them in any kind of pseudotime. Okay. So then the other thing to check, of course, is that, you know, if you have your ground truth, your annotated ground truth, you want to see how precise your method of classification is. So what was very nice about looking at a very aptly named confusion matrix here, was that, again, if we looked at the old classical way of doing the machine learning approach, you can hopefully see here that actually we've got some quite bad misclassification. Okay. But with the deep learning, we have a much better agreement between the predicted and the ground truth. Not perfect, but still very, very good. Okay, and then the other thing as well is that we found that not only did this deep learning approach allow us to pick out the major phases, but it also allowed us to distinguish between G1, S, and G2. Now, I don't know if you can see this, but we actually weren't able to do that using the classical. Oh, where was it? Yeah, so on the next slide here, on the classical way of doing it, we weren't able to distinguish our G1, G2, and GS from one another, and we had to kind of group them together. But Deep Flow was actually able to pull apart all 7 major phases of the cell cycle with, again, a very, very low false positive rate. Okay, so I want to summarize this part. So deep learning, for me, was incredibly powerful because you can develop and train a network in quite a short period of time. We can use it then to find our features for us and segment by itself potentially. And it seemed, in our way of doing it, very good at ordering cells into a class along this idea of a pseudo-time trajectory. My only concern always, though, that there's always this black box element to it. So, you know, we're never quite sure. We can look at the activation patterns at the end, maybe the network, but we don't know whether the features learnt may be linked to the biology or could be, as I said, potentially artifactual. And then, again, something that we probably don't worry so much about, but for rare cells like mitotic cells, there's always this danger that maybe there's an overfitting thing. Okay. So in the last 5 minutes, I just want to talk about what next. So I want to go back again to this idea of having to antibody stain your cells to identify who they are. So obviously you're quite used to working with antibodies against CD markers with a particular analyte on the back of them to basically tell us a little bit about our cell. But the question we're very interested in at the moment is, could we still ask those questions without labeling CD markers on cells at all, right? Remove the need for it. So again, this goes back to this power of bright field. So what I've got showing you here is 4 cells that have been imaged in bright field. And I can tell you that in a eosinophil, this is a monocyte, a lymphocyte, and a erythrocyte. So as humans, we're very good at looking at these images and seeing that qualitative difference. But what we want to do is we want to see whether there's hidden features, sets of features in here that could remove the need to stain with an antibody at all. So again, just to remind you, label-free is quicker, cheaper, and particularly with antibody binding, less likely then to cause an activation signal. So the way we're doing this at the moment is thus. So we're going to basically start work with human samples, whole blood potentially, but also mouse organs, lymph nodes and spleen. And we're basically going to either look at them in a resting state or look at different activation conditions. And we're going to put them through a fluorescence-based kind of semi-high throughput screen for CD markers, qualify this intensity staining on conventional flow cytometry using high throughput, and then migrate that information over to one of these imaging cytometer systems that also has a plate reader. So the idea here is that, of course, what we're going to have is that we're going to have not just the fluorescence information, but the bright field information correlated with every single CD marker in these different panels. Then, you know, this is the woolly bit here. We're going to work with several people to kind of mine into this data set and really see whether or not we can find features in the bright field which overcomes the need to even use an antibody. And then the idea is we're going to call this idea of a label-free CD collection. So the goal here is to identify cell phenotypes without a need for a CD marker. So let me give you one example, very, very simple, very quick example. So we found that Siglet 8 came up. So in terms of our little screen, we found a nice staining population on there. We then ran them through the imaging system, this particular hit, identified again single cells, pre-gated for focus, and then we extracted some bright field features. And what was nice is that when we visualized it using TSNE, there's about 200 bright field features in here. So it's not actually using deep learning. This is just using a form of basic machine learning, TSNE. We got some islands formed, and then there was this very distinct island here. So what's nice is because we have fluorescence information in the same data file, we can identify these fluorescent ground truth, and we can put it back. And there are our eosinophils. So we can clearly, in this case, replace the need to use something like Siglet 8 on a human blood sample to identify eosinophils. We can do this using bright field features only. So it was a good first start. And I'd like to tell you more about it, but we're still kind of chopping our way through the data. Okay, just to wrap up now, our final summary. For me, imaging flow cytometry is still one of the most data-rich technologies because it can analyze tens of thousands, hundreds of thousands of cells and get you 12 images per cell. And this really presents the single biggest challenge to how we interact with our data. We can do this in a manual, iterative fashion, but we know that this analysis is very subjective and poor in reproducibility. So we can use things like machine and deep learning, which should be much less subjective, give us many more features that we can look through with combinations, and hopefully, again, uncover features which we are not able to appreciate with the human eye. So this gives us this idea, again, of label-free possibilities using bright field, but also potentially using the scattered image as well. So we found in our hands with the cell cycle data that deep learning was superior to classical gradient boosting, but there was always this little issue about which features have been used to select the population. So as I said, we're very interested now in this. Can we refine or replace the need for CD marker staining using bright field features? And of course, we're also very interested in putting this into a clinical question. So I'm going to finish here. I'm going to acknowledge the incredibly intelligent individuals who have done this. I don't include myself in that. So I want to start off by acknowledging my lab. This is my team of cell detectives, as we now call ourselves. These are excellent people at the Broad Institute, Anne Carpenter, Min, Claire, Hugh and Paul, also from Swansea University, and Holger. All these guys were the people that really started this whole idea of using imaging flow cytometry and machine learning. I've already mentioned the work of the deep learning was driven by Alexander Wolf and Fabian Thies and Helmholtz, and then the idea for the data side of things really came from my time at the Francis Crick Institute or the Cancer Research UK in London. And then I'd also like to acknowledge Ivan Seis and Sophie van Gassen, who are going to be working on the label-free CD project with us. and the clinical links that I have at Newcastle University. So this is my email, this is our website, and this is our Twitter page. And finally, I'd like to say thanks again to the power of South American prayer. So thank you very much. Thank you. Thank you.
I have two questions. First, do you think that these bright field approaches will be able to identify very specific population and thinking of activation status of the lymphocytes?
So my feeling is I think it can. We do have some examples that suggest there are differences when you activate the cells versus when they're resting, but I'm not sure how deeply we will be able to go. I mean, could you find a CD4 T cell versus a CD8? Could you then find a regulatory CD4 T cell, a Th1, a Th2? I think we're in early days. I like to believe we can, but then I also realize the challenge that there has to be something then in the bright field, in the structure of those cells, which comes out. And I think that magnification could be an issue. We also have the ability to look at different magnifications on these systems, high, low, medium. I guess the kind of quest here would be, if it does work, you start to think about simple systems that take just bright field images of cells, telling you a lot about what they're doing. You can see that there could be point of care potential there. So I think that's the answer to the first question. A little bit woolly. I think there is a possibility, but really the proof will be in the data.
And the second one, what do you think about combining of complementary roles for transcriptomic studies and Cytometry? So, do you think that you can, in some situations, complement or they are kind of redundant information?
I think that, they're highly complementary because, again, transcriptomics tells you about the RNA. It doesn't necessarily tell you that the protein is expressed or whether or not it functions. So in the lovely piece of work that was done, that was published last year by Muz Hanifa, Anir Hakkanen and Alexandra Chloe Villani, the thing that I liked about the involvement that we had in that was that, we were part of this validating. So they found this new populations and defined them by our RNA, but of course then we use classical flow cytometry to redefine a panel of antibodies to select those cells and validate them by protein and validate them by function and by morphology. So I think, this is the thing, is that we mustn't be too led by single-cell RNA sequencing only. It's an amazing discovery tool and it will continue to be, but there's certainly a place for the lower dimensional technologies which validate those findings, because there are issues with single-cell RNA sequencing. There is dropout and bias, and a low-copy RNA may be doing something. It's everything. This is why I believe cytometry is a massive field. I'm a cytometrist. I'm not a flow cytometrist. I believe in integrated technologies towards, the question of single-cell biology, but in the context of the organism and of the population.
Are there...
Very nice talk, thank you. What do you think about these technologies for tissues when you disaggregate? Because the data you showed are blood cells, right? Does it work the same when you disaggregate tissue and you try to analyze that?
Well, this is my it's always a concern, So I showed in one of the early slides that we work with all types of cytometry technologies. So typically it's disaggregation of the cells, into cell suspension. But we now work in tissue cytometry. We have the mass cytometer that allows us to look at 40 antibody measurements on the tissue section. So I think this allows us to look at this question, whether or not you've changed something fundamental about the single-cell biology when you disaggregate a tissue. And we do a lot of tissue disaggregation. We work in gut and heart, skin. You always worry, what have I done? What's different? Have you killed a cell type? Have you stripped the receptor off so you can't see it? So I think the answer is that we're going to look at what we find in suspension, but we're also going to validate and look when we preserve the tissue.
By the way, what is tissue cytometry?
Okay, so tissue cytometry is cell measurement, and tissue is you're basically performing some kind of immunohistochemistry or immunofluorescence, or in our case it's metal tagged antibodies again. So we're metals, so isotope-tagged antibodies, CyTOF, right? So we have a system now which links into the CyTOF, which allows you to take a frozen tissue section slide or a paraffin-embedded slide and label them with maybe 38, 40 different antibodies that have metal isotopes on them, which can be read pretty much with distinction. And nothing's different. You do the same process as you would normally do, pretty much, and then you put it in this system, and a very high-power laser blasts 1 micron spots on your slide and samples the tissue into the CyTOF and looks at the isotopic composition. So what you're able to do then is, you get 40 directed measurements about phenotype. You get spatial resolution of your image, your tissue, because every pixel is then reconstructed into a pseudo image. So you get like a raster scanned image of your tissue. But then you can define phenotype by marker expression, by single cell morphology, if you can then segment the single cells in the tissue. But then you can ask about the neighborhood. You can say, well, cells of this phenotype, are they near cells of that phenotype, or are they far away? And again, this links with biology, because if you're a pathologist, that's what pathologists do. They look at tumor tissue, macrophage, T cells, where are they, So I think it's a really good question, and we're certainly aware of this idea that suspension is suspension, and I said, I think the other week, or this week, that no cell is an island. Even in blood, it's not an island. They come together, they talk, but.
Thank you.
Any other questions? I do have a simple one. Yes. I didn't see you.
Thank you for your talk. It was a great interdisciplinary work. I have 2 questions. First, about the technology. Do you use a standard CCD or a EMCCD detector?
It is a standard. I believe it's a standard CCD, but it operates in this time delay integration mode.
Okay.
So it has the ability, again, for the information to be passed down pixel row by pixel row as the cell travels. Yeah. But I say standard. I believe it's still a custom. It's a custom. I mean, the company knows how
many
it works.
frames per second?
I don't know.
Okay. Don't worry. And the other one is about the training of the deep learning network. Do you have an idea of how many images were used for training? How many images?
Events. Images.
Images.
Sorry, what events? Yeah.
Yeah.
I think we had something like 100,000 or something like this. Individual cells, and then, of course, associated images with those 100,000.
And the 300 features were all extracted from the bright field image?
Yes. Yeah.
You also add some manually tags to those
so
300.
Again, we had the expert truth
Yeah.
of annotating the data and defining what we see as true class based on DNA shape, DNA content, and a mitotic marker as well.
Okay, thank you.
Can I ask the simple question? No, in the approach that you have used for the cell cycle distribution analysis, have you tried, for example, senescent cell, how it locates? I assume that you will have a different, because they had a different size scatter, but have you ever tried?
We haven't. That's a really good idea, because finding senescent cells can be challenging. I mean, I know the size scatter will change, but people try things like Hirsch and pyronin staining, and that's not. We haven't done this. This is a really good idea. Yeah.
If there are no more questions, we thank you, Andy, again.